Hi, everyone. Welcome to our 3MT competition. We're very excited to have brought it back to campus this year. In previous years, we've had it at Pegasus Park, and we had a good turnout there. And so I think we have an even better turnout here on campus. And so thank you all for coming today. We have about 8 finalists who did amazingly to this point. And so happy to have them here so you guys can hear the amazing work that they're doing. We also have a great judging panel that Emmy's going to introduce you all to. So without further ado, I'll let Emmy go ahead and she's going to give a short presentation.
My name is Emmy, and I'm this year's 3-Minute Thesis Director. So to give a brief overview of the competition itself, 3-Minute Thesis is actually started from another university in Queensland, but it's now a universal global event where basically it challenges students to really think about presenting, communicating their science in a very efficient way. And the challenge is to do so under 3 minutes. And it's now practiced in many different institutions. So the main rules of this competition is that only a single static slide is permitted from beginning to end with no animations. No additional props are allowed, and all presentations are to be spoken. And the second that the presenter moves or begins to speak is when the 3-minute timer starts, so just be aware of that, and you'll have your timer in clear view in front of you. So the main judging criteria that you'll be evaluated on, the first half is of course on the comprehension and of your science. So is your presentation providing a clear motivation, background, significance of your science, and then ultimately the meat, which is your research strategy, how are you going to answer your main question, and of course the end, which is describing the conclusions, outcomes, and impact of your project. Sort of similar to many other scientific presentations we all give, it's just under the course of 3 minutes. And the second half of your judging criteria is going to be on your engagement and communication. How enthusiastic are you speaking? How well is your oration? Is the language appropriate for different audiences, not just experts in your field? That is something that is going to be a really important skill for you, not just for this competition, but just throughout grad school and beyond. And of course, the design of your PowerPoint slide as well, and just overall enthusiasm and excitement for your very own project. So the most exciting part, which are the prizes for 3-Minute Thesis. First place, which is determined by, of course, our panel of faculty judges, will win $500, followed by the runner-up or second place, which is $300, and third place, $200. And followed by that, we also have a People's Choice vote, which will provide an additional $100, and that will be decided at the end by a QR code that everyone can vote and choose their favorite speaker. So not just the judging panel, but also the audience gets to have a say. Now, I want to give a huge shout out and thank you to the judging panel today, Dr. Ravnik, Dr. Ripa, Dr. Kevin Forsberg, Dr. Prapti Modi, and Dr. Huiming Liu as well. Thank you all so much for taking the time to evaluate these amazing presentations. Thank you so much, and without further ado, I think we can get started. So first on the list is Anjana Sundarajan in the GDD program in Dr. Gary Hahn's lab, and she will be presenting the typos in TBX5 that break the heart.
Let me ask you a question. Can your heart break before you're even born? Most of us need at least a few bad life choices for that. But for one child born every 15 minutes, it happens at birth. They start life with a heart that's in a way broken and needs mending. Congenital heart defects are problems with how the heart looks and work, like this hole. They are the most common birth defect and cause more infant deaths than all childhood cancers combined. When I was shadowing a cardiologist at UT Southwestern, I met parents of newborns who were recovering from heart surgery. Parents asking, why did this happen? Could it happen again? Those questions lingered because surgery can repair the heart, but it cannot explain why it broke in the first place. And that's where we turn to genetics, because DNA, the blueprint of life, may hold the answers. But here's the challenge. Some DNA changes cause defect, but others don't. We often cannot tell which is which. We need a tool that can test these changes at scale and precisely measure their effect. So in our lab, we built a technology that does exactly that. Think of it like running thousands of crash tests, but instead of cars, we are testing changes in DNA and seeing if they have potential to affect heart development. We started with a key heart development gene called TBX5, one where some mutations are known to cause defects, but many remain a mystery. We tested hundreds of changes of TBX5 and saw a range of effects, but one result was striking: a cluster of mutations in a region, a spot where key heart building signals can come together, shown as red dots with yellow spotlight in the middle panel. So when this coordination falters, heart blueprint can be misread and can give us clues about how it is formed. And when we looked at this exact region for DNA mutations in healthy individuals, it was completely blank. Suddenly, the picture was clearer. This mutation was so important that it barely made into healthy individuals. So what? Today, when doctors find mutations in babies, they often cannot tell if it's harmless or dangerous. But technology like ours can help strengthen the evidence of which mutations likely matter. And it's also crucial for gene therapies. The first successful gene therapy treatment was only possible because scientists knew exactly which DNA region to look at, a feat possible by technology like this. My work brings that idea to the heart and potentially can be scaled to other diseases. So one day when a parent asks, why did this happen to my kid? We are one step closer to giving an answer and make sure the only broken hearts in the future come from bad decisions and not birth defects. Thank you.
Reagan Adams from GDD and Dr. Rachel Bailey's lab is next. And she is presenting Opening the Door to SLC 13A5 research.
Imagine a newborn baby, perfectly healthy one moment and then seizing within hours of birth. This is a reality of parents with children affected by SLC 13A5 citrate transport disorder, a rare developmental disorder where patients have severe seizures, intellectual disability, mobility issues, and require 24/7 care. SLC 13A5 makes a transporter protein. Think of this like a molecular doorway that brings citrate into our brain cells. Citrate isn't just part of your everyday orange juice, but it's actually a vital ingredient in things like energy, metabolism, lipid synthesis, and even neurotransmitter balance. When SLC 13A5 doesn't work because of mutations that actually break this transporter, citrate can't properly enter our cells. The result, we have too much citrate outside our cells and not enough citrate inside. Using a mouse model that develops the exact same disease as patients, I seek to understand how this citrate imbalance actually causes seizures. See, our hypothesis is that citrate could be disrupting how our brain cells regulate activity. Our brain does a pretty good job of keeping a careful balance between signals that excite and signals that calm activity. And we think that citrate might be binding to key regulators of this, causing a disruption in signal, which could cause seizures. In SLC 13A5 mice, I've been able to show that like patients, they have increased seizures and increased citrate floating around their brain. I've even taken this a step further and actually given these mice a diet of even extra citrate, and this has caused even further increases of citrate in their body and even further increases of seizures, which could suggest that citrate could be one of the culprits of this disorder, though further studies are needed. The goal, however, is not only to understand more about SLC 13A5, but to develop therapies that actually impact patients. Like most patients take between 2 to 3 anti-seizure medications with varying levels of control, creating a great need for better therapeutic options. In the lab, we use gene replacement as a therapeutic strategy. In this, we use a special harmless virus that actually encodes the instructions to make these functional citrate doors, and we can deliver these to the vulnerable cells. In mice, this has been shown to decrease citrate, be safe, and to improve seizures. And excitingly, this is actually entering clinical trials at UT Southwestern starting next year. So all in all, we hope that this research and this therapy will have transformative effects for patients affected by SLC 13A5. And more broadly, we hope that this research will open more doors for collaborative rare disease research everywhere. Thank you.
Next up on the stage is Dylan Calhoun in the Cancer Bio program in Dr. Garcia Bermudez Laboratory, presenting Fighting Fire with Lipoproteins, Antioxidant Defense in Cancer.
When I was a kid, my dad taught me the basic lesson of fire safety, given that he's a firefighter himself, to always know where my fire extinguisher was. I never thought this lesson would come back to inform my work as a cancer biologist, but it turns out that inside our bodies, our cells are fighting their own unique kinds of fires every day. You see, as our cells burn fuel to make energy, this biochemical reaction produces tiny sparks or flames inside of cells called reactive oxygen species, or ROS, that serve to cause damage inside of those same cells. If these flames or sparks go uncontained, that damage will spread in an uncontrolled chain reaction and lead to a dangerous cell state known as oxidative stress. Normally, though, healthy cells are able to avoid this outcome by using antioxidants, or natural fire extinguishers that work to stop these flames before they spread. Cancer cells, however, live under a constant state of oxidative stress, particularly as they spread through our body during metastasis or face treatments like radiation and immunotherapy. And when the flames inside of these cells become too intense, cancer cells are known to undergo a unique and iron-dependent form of cell death called ferroptosis, where the cell's membranes are oxidized uncontrollably. Essentially, the cell burns from the inside out. My thesis research set out to ask and answer a simple question. If cancer cells live under constant fire, how are they able to survive? We made the surprising discovery that tumor cells are able to survive because they don't fight the flames on their own. Instead, they call for backup. As oxidative stress builds up, tumors begin taking up lipoproteins from the bloodstream. Yes, lipoproteins like LDL and HDL, the same particles that we've traditionally known to transport cholesterol throughout our bodies. But these particles don't just carry cholesterol, they also carry vitamin E, nature's most effective antioxidant. In this way, you can think of lipoproteins as fire trucks that are delivering vitamin E fire extinguishers straight to the tumor. And by taking up these antioxidants, tumors are able to build a biochemical fire-resistant shield that protects their membranes from burning. In doing so, they're able to survive this incredibly dangerous environment that they live in. And in particular, when we deplete lipoproteins of vitamin E or block the ability of tumor cells to take up these lipoproteins altogether, this protection is lost and the shield collapses. Cells that once seemed highly fireproof become highly flammable. These findings have important implications for cancer biology, suggesting that the levels of lipoproteins might influence how tumors grow or respond to treatment. My dad always said that the best firefighting isn't about putting out the flames, it's about understanding how fires start, and I hope that this knowledge will help us extinguish cancer for good.
Well, up next is Jay Coronado in the immunology program in Dr. Tiffany Reese's lab, presenting the project Maternal Parasitic Infection May Shape the Next Generation's Immunity.
One of nature's most amazing yet mysterious relationships is that between a mother and her unborn child. Long before a baby takes its first breath, a quiet dialogue begins. Inside the womb, mother and baby are in constant communication, not through words, but through molecules. Nutrients, hormones, and immune signals pass between them to help guide the growth of every organ, including the immune system. The point of my project is to understand the purposes of what goes on between in the maternal immune system and in terms of a maternal parasitic infection. So using mice, I developed a maternal parasitic infection model to help test how well the offspring's immune system could handle a completely different pathogen, the flu virus. And what I found was nothing short of surprising. The offspring that were from the infected mother were far less susceptible to flu than those from the uninfected mothers. These mice lost less weight, stayed more active, and had completely healthier lungs. Meanwhile, the offspring from the uninfected mothers had lungs that were stuck in a constant state of inflammation. So these unexpected results gave us an opportunity to, moving forward, to identify the unique immune signals in our mouse model, demonstrating that we can further identify these and figure out what the problem is going on. This project is really great. I really enjoy this project. I do understand that this work is important because by decoding the maternal-fetal dialogue, we may one day support healthier pregnancies and equip infants with stronger immunity, thereby strengthening the overall health of both the mother and the fetus for generations to come. Thank you.
Up next is Malavika Jayaram in the immunology program in Dr. Neil Alto's lab, and she is presenting Stripping Salmonella, or STM, of its gear reveals immunological barrier.
So imagine a microscopic athlete trained to invade your cells and jump over barriers that's otherwise designed to stop infections. That's Salmonella, a bacteria so skilled it causes millions of infections globally. Salmonella, to me, is not just a foodborne pathogen, but a master strategist. It's able to produce these tiny molecular tools called effectors, which are like pieces of protective gear that allow it to hide, replicate, and survive. But the challenge in the field is that there are about 30 of these effectors, and it's difficult to pinpoint which of these effectors are truly important to cause a full infection in animal models. So my lab took a bold approach and decided to strip the Salmonella of its gear and see what happens. So on the left here, we have a wild-type Salmonella with fully equipped with its effectors. So every helmet, headgear, and shin guard in place. We went on to remove these effectors and call it Salmonella theta, which is like a athlete stripped down to his basics, waiting at the start line to face the immune defense. And this immune defense is interferon gamma, an immune signal required to restrict bacterial growth. However, when theta is faced with this defense, it's unable to pass through, unable to cross the barrier and cause systemic infection. So we went back and added a small set of effectors called SPV. And now our athlete, theta SPV, is able to cross the barrier and cause systemic infection. So this reveals something powerful. One, the interferon gamma is the key barrier that contains Salmonella, and two, the SPV genes help overcome it. So by stripping Salmonella to its basics, we were able to identify which barrier is important. And by further pinpointing which interferon gamma defense is targeted by SPV genes, we can reinforce this pathway to identify better therapies. And this is important because Salmonella is very much a global health threat, especially in regions with low access to clean water, as well as antibiotics. So next time you think of Salmonella, don't think of a bacterium. Think of a world-class athlete racing past immune barriers. And thanks to our minimal genome system, we're now able to figure out which barriers and how to stop it before the finish line. Thank you.
Up next is Sarah Jalvins in the neuroscience program in Dr. Helen Lai's lab, and she'll be presenting her project, Pain Sensing Neurons, Assembly and Care Instructions Inside.
Although millions of people live with acute and chronic pain, there are very few effective and non-addictive treatment options available, partially because we don't fully understand how pain-sensing neurons are developed and maintained. This is the question my research tries to answer, with the aim of trying to help development of new pain therapies. To help you think about how we understand this, imagine that you're building some IKEA furniture, perhaps a desk and a couch. You'll probably want unique instruction manuals so you build them correctly, right? Now replace each piece of furniture with a unique class of sensory neuron and the manuals with unique gene combinations, their little hats here, and you'll have the basics of how we hypothesize sensory neurons are generated. In this example, the desk is a nociceptor, or a pain-sensing neuron, and the couch is a proprioceptor, which provides our sense of limb and body position in space. We got a clue about how these sensory neurons obtain their identities from a disease called Puridium-12-associated congenital insensitivity to pain, in which patients have a mutation in a gene called Puridium-12 that results in their inability to feel pain and subsequently frequent injuries. When we recapitulate this in mice by removing Puridium-12 before birth, we see that pain neurons fail to develop and that the animals don't feel pain, indicating that Puridium-12 is critical for the development of pain-sensing neurons. However, proper maintenance of neuron identity is also important for proper neuron function. And so I wanted to know if Puridium-12 is also important for maintaining the identity of pain-sensing neurons into adulthood. To answer this, I removed Puridium-12 from adult neurons in mice after normal development has occurred, and then explored the expression of various genes that we use to identify specific types of neurons. In doing so, I found that we get expression of a particular gene that we use to identify proprioceptors appearing within pain-sensing neurons. So what does this mean? Essentially, under normal circumstances, PRDM12 is actively preventing gene markers for other cell types from appearing within pain-sensing neurons, allowing them to maintain their unique identities. However, without PRDM12, we get neurons that are a little confused because they have two different identity markers. It's as if someone were to hand you a manual that's half A and half B. You'll wind up building something that resembles half a desk and half a couch. This is especially interesting because it suggests that one gene can control both the development and the maintenance of neuron identity simply by determining whether it provides the assembly or the care instructions. Moving forward, we hope that understanding these developmental and maintenance processes will allow us to be able to grow and regenerate pain-sensing neurons in order to help provide new pain treatments. Thank you.
Next is Aliyah Shed in the neuroscience program in Dr. Kimberly Huber and Dr. Jay Gibson's lab, presenting sock seams and sensory storms in Fragile X syndrome.
So, you know the seam on the top of your socks? If it's just a little bit out of place, it really bothers me. Now, imagine if that's how you felt about all of your incoming sensory information. Fortunately, our brain filters out billions of distracting information, like our socks, so that we can focus our attention on what's important. But for people with Fragile X syndrome, this filter seems to be impaired, and that can make them extra sensitive to sensory information and can contribute to sensory hypersensitivity. So Fragile X syndrome is one of the most common causes of autism, and it is marked by this sensory sensitivity. So while hopefully your socks aren't distracting you from listening to the most important thing, me, someone with Fragile X syndrome could be overwhelmed by all the extra sensory information because their brain is struggling to filter out all of the information. So to understand why that happens, for my work, I'm looking closer at how brain cells communicate to process sensory information. We have our go cells, which increase activity, and our stop cells that shut it down. And for them to communicate, they work through different types of receptors that work like gates, but not all of the gates are easy to get through. So when the information comes in and opens the gate to a go cell, there's also this roadblock. So it has to be strong enough to move that out of the way first. But in our stop cells, they have receptors that don't have this block. Now, through my work, I've discovered that some of these receptors are actually working for the wrong cell types in Fragile X syndrome. Why is this important? Because this receptor organization is important to maintain a balance of activity, to keep our information processed properly. But in Fragile X syndrome, the balance is shifted towards increased activity. So with these unblocked receptors in the go cells, they become way more chatty, and the stop cells can't keep up. But we can rescue this by using a drug that specifically impairs these unblocked receptors, and also by preventing these unblocked receptors from ever working from the go cells in the first place. So this is a new piece of the puzzle in understanding the function of the brain in Fragile X syndrome and what contributes to this increased sensitivity to senses and increased brain activity. And hopefully this work can contribute to creating specific treatments that don't exist for people with Fragile X syndrome. And maybe your sock seams don't have to create sensory storms. Thanks.
Last but not least is Cody Tufti in the BME program in Dr. Mohammed Rizwan's laboratory, presenting cornea glue. Don't just seal, heal.
Now, imagine you have a daughter named Christy, and one day she's playing in the sand, and you hear a terrible screech, and she's in tremendous pain, so you take her to the hospital, where a doctor there tells you that a grain of sand has lacerated her cornea, and scarring will soon steal her sight. Now, corneal scarring is one of the leading vision-related disabilities worldwide, and our only retroactive treatment for it is a cornea transplantation. It can be caused by a variety of ocular diseases such as cataracts, fungal infections, or the like. Now, with transplant surgeries, there is of course the limitation of donor tissue shortages. And in the case of the cornea, there exists less than 1 cornea per 70 victims. This means that our surgeries and recoveries have to be precise and done with care. Now, within corneal surgeries, there perform lacerations along the periphery of the cornea that are later sealed with sutures or the like. Now, as we see in the bottom left, sutures can cause additional damage to the cornea and stimulate their own fibrotic response, counteracting the surgery we just performed. Additionally, commercial sealants such as fibrin glue seek to aid this, but because they focus on sealing the cornea, they inhibit infiltrating cells, which increases the risk of transplant rejection. Now, my research seeks to develop a corneal glue that doesn't just seal, but also heal. To do this, I've devised a two-pronged strategy with enhancing cell infiltration and repopulation of the wound, as well as reducing the fibrotic process to facilitate scarless wound healing. To increase cell infiltration, I engineer interconnected porosity by fabricating gelatin hydrogels, or water-based gels, into microspheres and jamming them into a granular hydrogel as seen in the SEM image in the top right. Now, similar to volleyball stuffed into a bin, there exists an interstitial space between each of our microgels that allows cells to migrate into and throughout the scaffold with little to no resistance. When placed in the cornea, the scaffold also becomes an artificial aid to regenerate the lost tissue. Now, the fibrotic process within the cornea mainly consists of our resident cells being transformed into inflammatory myofibroblasts. By engineering the sustained release of the anti-inflammatory cytokine interleukin-10, I aim to block transforming growth factor beta, which is primarily responsible for cell transformation. And I can engineer this within my adhesive glue by simply subbing out subpopulations of microgel within the scaffold. The combination of these two strategies seeks to create a surgical sealant to help little Miss Christie get back to her days and restore her vision. Thank you.
Thank you to everyone again for participating and for a great 3-minute thesis round. So we're going to start with the People's Choice vote first. Drum roll. For People's Choice, it's Reagan Adams. So huge congratulations. We'll start with the judges' choice. So in third place is Malavika. Awesome, great job. And second is Sarah. Good job. And finally, the first place winner is Dylan. Congratulations. Congrats to the 3 winners. Seriously, like, amazing job by all of you. But seriously, to all the participants, amazing job. Thank you so much for working hard and giving your talks. Thank you again to the judges for your time and everyone else for attending. And thank you so much for making 3-minute thesis again happen this year. So thank you guys so much.